He started his electrophysiology research at the Communications Biophysics Laboratory here. So this is a connection for him to NIH. And he received the degree of Doctor of Science from George Washington University, and for the past 30 years he has been involved with neuropsychiatric research at the NIMH. And in particular, he is now the director of the NIMH MEC core facility and has pursued many types of studies there, especially related to cognitive behavior and brain dysfunction. And today his title is MEC and neuropsychiatry. Please.
Well, thank you very much for inviting me. This has been a really wonderful pleasure to come back here, as Matti said, in many ways. While David was down the street at the Magnet Lab, I was in one of the Quonset huts just a few yards away from here in Building 20, doing my dissertation with something called the ARC, the Average Response Computer. And this was Professor Clark had built the first sort of transistor digital CAT, the Computer of Average Transients, except it was 3 racks of equipment that we reeled around and had switches to kind of program. And I was doing mapping, not mapping because we had one little electrode stuck into into a rat that we were trying to look at auditory heart rate conditioning. So that was, and this whole area was quite different. Kendall Square had quite a different character. And there's also another sort of interesting synchrony of these things, because in 2002, when we brought up the MEG lab at NIMH, Bob was our scientific director then, so he was my boss back at that time, and was really very instrumental in helping bring that lab into being. Back at, it took some number of years, given the government exigencies of how you raise funds and get things together. But we were finally going in 2002, so that was very nice. So what I really want to talk about today is follows on to the last talk. We're moving into sort of applications and into I'll use all the current buzzwords like translational. And I've always viewed myself as sort of a translator, because I was here at MIT in the electrical engineering department. We didn't have neurosciences, or what didn't exist then, and neither did the brain department, any of these things. So to study, what I was interested always in studying the brain, and the only place that was sort of doing that as an information processing device was the Communications Biophysics Lab here. So that was sort of how I started. But that gave me the sort of the background about some of the methodologies and the things that you need to do to study information processing in the brain. And then on the other side, to be able to also speak to the clinicians who were interested in trying to use these things, not just the clinicians, but also the people doing basic cognitive work. So that's the way I use the word translational, which is a little bit different than the way they do now. But in early days at NIMH had been interested in looking at individual differences. And so the way is, how does those individual differences translate into the search for biomarkers and intermediate phenotypes? And those are the other two big buzzwords that we use. And they come very much out of some of the kinds of things that have been driven by genetic research programs into some of the background of mental illness, in that we can study the diseases themselves as a category, which we can do and get categorization that way. But many of the diseases, qua disease, don't travel necessarily as their own sort of genetic component. But the disorganization and the symptomatology and the other pieces that go with these diseases are the things that we're sort of after. They can give us not only diagnostic things, but hopefully treatment targets. And that's really sort of where some of this comes from, why we use the terms like biomarkers and intermediate phenotypes. And I'll explain a little bit more as we go along. So this is our machine. It's what had been at the time the CTF machine. This is a 275-channel axial gradiometers. It has a set of 29 reference channels, which it uses to give you good noise immunity. We're in a vacuum schmelzer 2-layer room. I won't belabor too much of that. In terms of the methodology, I'm going to stay away from that. But one of the very first things we did was really want to get into the notion, can we do functional localization, functional mapping in very similar ways to what at that point in time we had been used to doing in fMRI. And so I'll just describe sort of the first set of things that we did way back when, which was to help us understand that we believed we knew what we were doing with the MEG in terms of these kinds of localizations. So we started out with something called the NBAC task. We were interested in working memory. This is a thing that the clinical brain disorders branch and Dr. Weinberger and other groups had looked a lot at in terms of fMRI and schizophrenia and other things. We wanted to see, could we follow this in MEG in the same way. And I'm going to use this first experiment as a way of sort of just giving a general overview of the kinds of methods we use. They're not specific. They're not the exact same methods across all the studies that I'll show, but they'll give a general time. The working memory task is pretty simple. You're seeing stimuli. You're either responding to the exact trial you see or one back, and so we can modulate the load of the working memory. This should be pretty familiar. Our MEG processing is pretty straightforward. Most of what we do is a beamforming technique. This has been the CTF machine and the people at that group had been promoting this as part of their beamforming technology. It's an optimum spatial filter. We use in this task. The task was blocked with the 2-back versus the 0-back control tasks, much the way we block things in AB designs in fMRI. We were still in the frequency domain at that time, so there's the letters there. Our data is grouped using everybody transformed to Telerak atlases. We use AFNI. It's very nice because Bob Cox is upstairs from my office, so that helped in a lot of things. And we'll be looking at changes in power that we think as either desynchronization, which is a reduction, or read as a positive. Excuse me. Got to get this to go the right direction. Our group analysis is, again, as I explained, is normalized by doing everything in AFNI. We can use a 3D t-test to threshold our group mean map to look for significances. In our fMRI, since we started out with the comparison to fMRI, so these are the very first data I'll show is just the same subjects doing the same task in the fMRI. This is very standardized SPM processing, nothing unusual. At that time, it was SPM 99 because we were back before some of the current versions. So the single subject is a block design contrast for this working memory load, and then the second level contrast, which is the group data. If you do this and compare your sort of group activation maps, these are both thresholded out at a corrected value of about P001. The color scales are a little different because they came off of the two different processing streams. But in this case, what we see is dorsolateral prefrontal cortex on both sides and posterior parietal cortex on both sides. We see very similar pattern in our MEG. In this case, it's beta desynchronization, which is sort of the hallmark that many people have shown as a correlate to the bold signal. It's not that we're trying to duplicate bold or trying to make the case for the same thing, but this was to give us the idea of that our functional activation and localization was comparable. In this case, it's a desynchronization, as I mentioned. If you change your threshold levels, these maps can become very much more similar. If you relax things here a little bit, you pick up this spot over here. If you tighten this down a little bit, it breaks into two spots very much like this. We can do individual level comparisons like this. One of the things I've always been interested in and what we want to drive from this is how well we can do individual differences. This is a single subject's result, which looks very much like group data for a single subject. This is the bold fMRI for that same subject. So even on a single subject level, we can get, this doesn't always come out this way, but that's one of the ones where it does. Well, how do we apply some of these things? For those of you who know the clinical brain disorders branch in Danny Weinberger, they've probably seen this slide a million times. But the idea to his study of schizophrenia was that we weren't really looking at schizophrenia, quasi-schizophrenia, but rather looking at the behavioral phenotypes and trying to figure out how did these things come about from a neurophysiological point of view. And so those are the kinds of things that motivated the design of the sibling study. In this case, this was looking at schizophrenic subjects. They're well siblings, and a large group of normal controls that were matched to all of this. Again, by just sort of going through this by way of trying to motivate the early parts of our data and why we were interested in how we get to this notion of intermediate phenotype and the idea of biomarkers. So again, we weren't interested in investigating cognition via this working memory task across these three groups. And I'll just summarize quickly on that just to get to the group maps. So this is that same NBAC task processed in a very similar way. Our normal controls, this is a fairly large group of subjects so that we get fairly large significant areas. This is sort of, if you've read Friston's recent ironic rules about statistical things, you realize that if your groups are too big, you can get things that are more significant than you might imagine. But that's why these blobs become very big, because we have a very large group. So our statistical significance gets sort of inflated to some extent. The schizophrenics show a lack of activation in the DLPFC. This is sort of no surprise. This is part of the notion of frontal executive dysfunction in schizophrenia. The thing that we were interested in particular here was the unaffected siblings, which show an intermediate physiological dysfunction in between the normals and the probands. And this is sort of what we meant by an intermediate phenotype. And then this gives us now a target that we can go after in terms of looking at the genetic background and trying to dissect things relative to the disease. That's sort of the intermediate phenotype biomarker motivation. We can start that kind of the kind of quest for looking at genetics behind this by trying to think about, we take sort of a candidate gene approach in this case, which was to what things affect frontal lobe function and working memory. Again, many of you have seen this if you've been through some of the CBDB type talks. And so COMT, which is co-methyltransferase, which metabolizes dopamine in the cleft. And there's a functional SNP. There's a single nucleotide polymorphism that gives you two genetic variations, two alleles of this that have an enzyme that are functional in the sense that they have a difference in the enzyme activity, and they will differentiate the speed and metabolism of reconstituting the dopamine. So this is a candidate for things that interact with the dopamine system in the frontal lobes. So we can begin to dissect the kind of physiology data that we're getting out of the MEG and bring our genetics to bear on this. So the SNP that modulates this is a Val-Met polymorphism, and so you wind up with three alleles for this: Val-Val homozygote and the Met-Met and the Val-Met. The enzymatic activity has been shown to be on a continuum across these. We were a little surprised by what we saw here because we were expecting to see a continuum across the three groups. Instead, what we found, and this has been borne out in some more larger data, more recent group, is that the Valmet looks like our sort of normal group map, but the MET-METs and the Valmets both show reduced frontal lobe activity. This isn't quite as dramatic as the programs itself. It's because the significant plotting here is a little bit different to sort of enhance the distinction between these, but it winds up being an inverted U curve in terms of the functional behavior. So that sort of all kind of background. These are studies that have been around for a while. We can also look at the resting data, which is again almost it's sort of an oxymoron of our notions of cognitive of science, because what does a subject do when you say relax and stay in the scanner? And that may in and of itself represent a different task to different clinical groups. So I'm always a little cautious with that. But we can even with that we can localize across our alphabet soup of rhythms. In this case, we see gamma-powered differences in precuneus, posterior areas for schizophrenics in the same condition. Now, whether or not this is due to a very different experience of that group in the scanner or not is an issue, but it gives you a starting place to begin to look at some differences. Now, we were interested, I shouldn't say we, I have to explain a little bit how our lab works, is that it's a core group, so their facility and many investigators throughout the intramural research program, both in NIMH and in NINDS, utilize it. So there's a quite a large number of users that bring quite a bit of background to the kinds of questions they ask. So this was a neuropsych group that was interested in dissecting our working memory findings, and this was using a Sternberg task. They were interested both in can you now try to dissect the pieces of the working memory. The Sternberg is as many probably know, is you're presented with a memory set. There's a delay period and then a probe as to whether or not something was in the memory set. So you've got an encoding phase, a decoding phase, and then a recall phase. And so you can look at different areas and pull out different time courses that represent what's going on. So this allows us to look at utilize the dynamic ability of MEG to dissect these pieces in a way that's a little harder to do with fMRI. If you take those time courses from the different pieces and you can look at what's happening. This is during this is during the intertrial interval, and we see our same frontal areas, but now instead of a decrease, we're seeing an increase. Now this is intertrial, so this is sort of a rebound of or a preparatory beta for what's going to be needed coming up during the task. In the delay periods itself, these are three separate sort of pieces of the delay. We see the encode, which is very visual because the data is being presented visually, and this represents our activation. This is the rebound from that, and then the frontal areas come online. And then finally, during the retrieval period, we see similar networks to that we do in the working memory. So this just is an example of the way we can kind of sequence through the time and dissect this. But actually, one of the things I wanted to bring out from that study by Mario was the notion of individual differences and how those relate, because that's ultimately what we're going to be interested in terms of biomarkers and clinical applicability. Is we know we can sort clinical groups. I mean, as as some have said, you don't need a diagnostic test at this kind of level to determine whether someone's a schizophrenic or not. You just need to look at their fingernails. Maybe not so true these days because smoking's a little bit down, but they will see the nicotine stains. And you can, so we don't need these elaborate tests for that. What we want these for is to be able to look at individual differences and make distinctions about them. This is the beta band activity from the left DLPFC during the preceding trial onset. So this is the preparatory stuff that's going on relative to this subject warming up to be ready to do the Sternberg task. In other words, they know they're about to need to memorize the things that are coming up. And these are the different subjects. Now, everybody has some beta increase during this period. Now, whether this is rebound from the previous one or is a true preparatory thing is a little bit questionable. But the point is that every subject shows this piece. If we collapse across time, we see this area. Now, not everybody's timing is exactly the same. But everybody does show a peak at some point during this preparatory interval. This is one of the earlier. David Coppola mentioned about the timing is everything, but sometimes we don't always know when the time is, for long tasks like this. And so we need to have ways of kind of sorting that stuff out. But the idea here is that we see a good deal of intersubject variability in this latency, but yet there's a substantial consistency that represents what the brain has to be doing. And this is just another example. We can take this is using our beamforming technique, but now on a time sample basis to pull out the network and the pieces of it on 50 millisecond slices that are 100 millisecond slices that are slid by 50 milliseconds across. This is for a single subject. This is just to illustrate, again, the timing differences. So these are three different areas, 8, 9, and 4, 5. So these are near the DLPFC and nearby. And these are the timing for those. These are from 20 different subjects. And there's again, you can see the activations, which are given by these decreases, but the timing is not at all identical across this because our version's working memory task, there's an explicit button response the person has to make, but the working memory involves sort of updating your internal scratch pad, but that's over hundreds of milliseconds and is not something that's going to happen at a punctate point. So again, the notion of individual differences and how we hope we can exploit this in some of our MEG models. Well, we had other groups in the intramural program who also wanted to take sort of a candidate approach, but not now in genes, but more in a candidate of areas of the brain that were their favorite spots, and they wanted to look at affective disorders. So this was a group in the mood and affective disorders. And one of the first things, of course, was to prove to themselves, just as we had wanted to see, do we get functional localization similar to the fMRI? They were particularly interested in, because of affective disorder, were interested in could they see amygdala activation? And this was an early experiment by Brian Cornwell to repeat Ahmed Harari in the clinical brain disorders group had done a face matching task comparison where the task is to match the face, pick out the matching face. The control task is to match the shape. The emotion is not the task here at all, but the comparison, because these are emotional faces, and in fMRI you get very clear-cut amygdala activation. He was able to show that you could actually see the amygdala given the right design and the right beamforming techniques, because what we had shown before was very much sort of cortical activation, and people were happy with that, but whether you could see things at the level of the amygdala and the hippocampus was questionable. So that was his early work to try to demonstrate that. And he also showed that, and by developing a sort of a human variation of the Morse water maze, could get very good hippocampal activation signals with MEG. And then he and And the group in the mood and affective disorders group wanted to apply this to looking at people with affective disorders, and this was also a preliminary to some of the ketamine studies. But this was to show that that he could see hippocampal, that the normalized theta was very, was highly related to the latency. This is how long it takes you to accomplish the navigation task. But the ultimate sort of culmination of all of that was to use a combination of the working memory tasks and these navigation tasks to look at the application of ketamine as an antidepressant in mood and affective disorder. This was really not pioneered by, but a major approach of Carlos Serrati and his fellow Giacomo Salvadori. And this was a paper that back from 2010, and this is sort of where we come up with the notion of a biomarker. So this was looking both at the beta power in desynchronization. In this case, it was a correlation between beta in anterior cingulate, predicted the degree of change in affect from the short-term application of ketamine as an antidepressant. And so this was the difference in the beta score, beta coherence, and this was the difference in the coupling. And this was, again, really showed the ability to have a predictor of antidepressant, which could then be also used as an outcome. This was there was even an editorial about this, the notion of sort of just to get the buzzwords, and this is how I come back to translation. All this isn't how I consider what I do is translational, but that's just helping talk between them. But this was the notion that with these kinds of stress tests is a way of being able to get a better glimpse into who will or who will not respond. And I think that's one of the really potentially tremendous application areas that we have access to. Going to finish up with some more notions about networks, because that's the other area that we've been really keenly interested in. Danny Bassett, who had been a postdoc in our lab quite some while ago, has been one of the patients looking at applying the sort of notion of graph theory, which is very much in vogue. This was looking at the efficiency of beta band networks in terms of being able to predict accuracy in our NBAC task, so that you can take these small world graph measures, which you develop from functional coupling patterns across brain regions, as a way of being able to detect not just the patterns, but to predict that the network efficiency and the network characteristics relate clearly to behavioral things. And this is, I think, as a follow-on to Professor Georgopoulos' talk, where the network issues are very key to the disorders. Here we have showing that the networks, at least as elucidated by the graph methods, really relate to the people's ability and performance levels. And one of our visiting fellows in our lab the year before, Shannon Weiss, took this idea a step further, because he wanted to look at could we actually modify and make changes in these brain networks. In other words, how much plasticity was there? And this was one of those sort of rare things. He was going to be a visiting fellow for a few months, and he said, "Well, I want to learn how to do MEG. I want to train schizophrenic patients and see what their plasticity and do." And I said, "Well, you're talking about a 3-year project. But Shannon turned out to be one of these wonderkins who was actually able to do all of this in about a 4-month stint in the lab. Partially that comes from the ability to have total focus on one thing and not be distracted by administration and other things. But basically, what he was doing was looked at network brain measures before and after auditory training. It was a very simple task. This wasn't aimed at cognitive remediation per se in schizophrenia, but it was sort of more of a pilot study than anything else. But subjects it was a tone discrimination task. People were studied before, with a baseline MEG, and then they were trained in multiple sessions. And then studied again afterwards. There's clear, nice behavioral things that pre and post, both the controls and the probands showed an improvement in their actual, fractional correctness. And this follows a curve over time. The shorter blue lines here it was a little harder to get the schizophrenic patients through as many sessions, but that's part of the rigors of that kind of study. He was just showing the correlations between task accuracy and these graph-constructed metrics. In this case, he's looking at the beta band, making measurements of both positive and negative correlations between brain regions, and how that relates to the score. So this just shows that you have a basis for believing that this again predicts accuracy and behavior. And then was then looking at the changes in these brain networks after the practice in both controls and probands. And so these are the areas that showed significant increase in connectivity that related to the performance. So this was a very pilot study. There's a lot of sort of issues to be dealt with here. We were looking at both beta and gamma. We were a little surprised at how similar these were. Whether that's sort of a more larger artifact, I don't know. But it was very promising in the sense that now we've got an outcome measure that we can show that the kind of plasticity that we see behaviorally in what could be possibly cognitive remediation tasks, we have a way of tracking using changes in network behavior in the brains. So. There's a lot of other things going on. This is my flashy picture. I had to add this in because you got to do it. I mean, one of the things that we are so captivated by MEG for is the fact that it is dynamic. That this is an auditory task. This is about 1 second of brain activity. It's slowed down a little bit. This is looking at gamma band activity. We can see some differences between right and left hemisphere. It was an auditory discrimination task. So there's some small language component to this. But this is our challenge, which is how to reduce. I mean, this is one of the reasons why I'm excited to sort of be at MIT and to see a thing here, because one of the things, this is signal processing problems. I mean, there's a lot of stuff to be done. I think Bob alluded to it early this morning, that there's just a lot of room left. I mean, the instruments are now very good. Compared to what we had, 15 years ago, where you had to, take a 1-channel or 7-channel or even 37-channel and move it around and all the noise. The whole head machines are reliable, but we've got to have a way of taking this dynamic pattern and reducing it and figuring out how to do it. We've got, there's questions about how you do the network connectivity. There was a nice discussion, pre-whitening or not. I mean, there's all rhythmic issues, that are basic questions in the methodology. And it'd be nice to see, a place here at MIT where you can really, hopefully attract some of your really powerful signal processing people in to be able to look at to look at kind of this. This obviously, as somebody said, it takes a whole lot of people. I run the core facility. I've got 3 staff sort of scientists who help with the analysis and help our subjects. Most of the studies come from other labs in the intramural program. Mood and affective, this was, I've mentioned some of their names already, the neuroimaging group within the clinical brain disorders branch, and Dr. Weinberger has since moved on to the Lieber Institute, but still very much, the motivation behind the SIB study. There's a lot of other things going on in the lab from many other groups that I didn't get to. A lot to do with resting connectivity, a lot to do with looking at other relationships when studying GABA spectroscopy and showing correlations with gamma activity. There's a lot of people doing in Mark Hallett's group and Leo Cohen's group looking at biofeedback using MEG-derived real-time beamform signals from very specific locations to help with stroke rehabilitation and brain communication interface work. There's a poster here from member Jabi who's looking at a dynamics processing stream in emotional faces, which I urge people to take a look at. So there's a whole host of things. This is just a little tip of the iceberg of the kind of flavor of what is possible, I think, in cognitive science and particularly in neuropsychiatry. So I think this is a great spot here to have machines set up, and so very enthused to see that there'll be a lot of outcome from the lab here. Thank you.
Thank you for this exciting presentation of many new possibilities.
That's from the last BioMag, next BioMag this summer. Just to give a little, sorry.
So, time for questions.
Quite technical question. Quite early on, you mentioned that you base your beamformer on I think you called it on the slide, dual state covariance matrix.
Yeah.
That means you pool the 2 conditions into the covariance matrix that you want to compare.
Well, there's 2 ways to do it. You can either pool it or you can do them separately. Either way, and then maximize the difference between the two covariance matrices. That's the way to give you your spatial, separation. Yeah.
Kind of combine them.
Yeah, it depends on the way the task was designed. What makes the best sense in terms of how to do that. I mean, this is something that people are looking at in terms of what's the best way to get your beamformer weights. You want to do all of your data and then project it back through the time course, or to try to get your weights from separate pieces. So, depending on how your experiment is set up, whether it makes sense to pool your covariance first or to break it up into pieces, get the locations, and then just project the weights out over time. But those are, I didn't really want to go into all the methodological issues, but that's a very important one, and that's one of the places where there's a lot of work to be done. Actually, one of the places where we're doing, which I'm kind of excited about right now, and that was sort of why I put this up a little bit, was that really the registration between your MEG and your anatomy is sort of an important one. And the better you can do that registration, the better you can have your forward models for doing this kind of work. And so we're working now on not using an actual brain hull model with the MEG data itself to decide the registration, so that you can get it very much better, tighter than using fiducial marks or some of the other things. I think that would be a helpful step in the kinds of things we need to do.
Okay, if not, we thank the speaker again. He started his electrophysiology research at the Communications Biophysics Laboratory here. So this is a connection for him to NIH. And he received the degree of Doctor of Science from George Washington University, and for the past 30 years he has been involved with neuropsychiatric research at the NIMH. And in particular, he is now the director of the NIMH MEC core facility and has pursued many types of studies there, especially related to cognitive behavior and brain dysfunction. And today his title is MEC and neuropsychiatry. Please.
And I've always viewed myself as sort of a translator, because I was here at MIT in the electrical engineering department. We didn't have neurosciences, or what didn't exist then, and neither did the brain department, any of these things. So to study what I was interested always in studying the brain, and the only place that was sort of doing that as an information processing device was the Communications Biophysics lab here. So sort of how I started. But that gave me the sort of the background about some of the methodologies and the things that you need to do to study information processing in the brain. And then on the other side, to be able to also speak to the clinicians who were interested in trying to use these things, not just the clinicians, but also the people doing basic cognitive work. So that's the way I use the word translational, which is a little bit different than the way they do now. But in early days at NIMH had been... Well, thank you very much for inviting me. This has been a really wonderful pleasure to come back here, as Marty said, in many ways. While David was down the street at the magnet lab, I was in one of the Quonset huts just a few yards away from here in Building 20, doing my dissertation with something called the ARC, the Average Response Computer. And this was Professor Clark had built the first sort of transistor digital CAT, the Computer of Average Transients, except it was 3 racks of equipment that we reeled around and had switches to kind of program. And I was doing mapping, not mapping, because we had one little electrode stuck into into a rat that we were trying to look at auditory heart rate conditioning. So that was, and this whole area was quite different. Kendall Square had quite a different character. And there's also another sort of interesting synchrony of these things, because in 2002, when we brought up the MEG lab at NIMH, Bob was our scientific director then, so he was my boss back at that time, and was really very instrumental in helping bring that lab into being. Back at, it took some number of years, given the government exigencies of how you raise funds and get things together. But we were finally going in 2002, so that was very nice. So what I really want to talk about today is follows on to the last talk. We're moving into sort of applications and into I'll use all the current buzzwords like translation, interested in looking at individual differences. And so the way is, how does those individual differences translate into the search for biomarkers and intermediate phenotypes? And those are the other 2 big buzzwords that we use, and they come very much out of some of the kinds of things that have been driven by genetic research programs into some of the background of mental illness, in that we can study the diseases themselves as a category, which we can do and get categorization that way. But many of the diseases, qua disease, don't travel necessarily as their own sort of genetic component. But the disorganization and the symptomatology and the other pieces that go with these diseases are the things that we're sort of after. They can give us not only diagnostic things, but hopefully treatment targets, and that's really